It's a real pleasure to be here. I'm looking forward to the rest of the meeting as well. And so, yeah, I want to tell you about morphogenesis in flowers. And oops. So one of the questions that we're deeply fascinated by is, how do organs form with consistent sizes and shapes? And this is a fundamental question that applies to basically all multicellular organisms. As you can see, we've got these nice Arabidopsis flowers here. They're sepals, which are these outermost green leaf-like organs. Each flower has 4 of them, and they're all the same size within a flower, and they're all the same size across flowers, so very reproducible or robust. You can think about mice, and their 2 cute little ears are both the same size. And you basically never see something like this with big and small sepals in the same flower, or a mouse with a giant ear. So this is a fundamental question of how do you have this robustness of organ size? Not only do sepal sizes need to be robust, they need to be robust throughout their growth, which makes it an even more challenging problem. So what sepals do is they're defensive organs, and they're protecting the inner reproductive organs while they're developing. And so the very first thing sepals do is grow and cover that flower bud. And so we're looking down on an Arabidopsis inflorescence, and you can see the developmental progression. So the youngest flowers are here in the middle where they're produced, and then they get older as you go outward. So if you get way down in here, you'll see some open flower buds, but they're below what I can see here. And you can see that even these very young flowers, the 4 sepals are closing that bud, keeping it protected. And then they undergo this massive amount of growth to mature sepals, and all that time, all 4 sepals are the same size. So they really have to maintain this robustness throughout growth, and that makes it an even more challenging problem. So what we love to do in the lab is live image and watch this process as it's happening by putting living, usually inflorescences, on the microscope and imaging them every few hours. And here we've got a fluorescent marker for the plasma membranes and fluorescent marker for the nuclei. And you can see there's a lot of complexity going on with those cells. Okay, so here's a sepal, here's a sepal, here's a sepal, and there's one in the back we can't see. And you can see during all that growth, that flower bud is staying closed. This is about 66 hours. Ooh, what's this? Okay. I don't know what this is. Let's get rid of it. Ooh. No, I can't even get there.
I think this is it.
Oh, it's over here. That's the problem. Let's just close it. Okay, there we go. Okay, so yeah, how do plants... So you can see lots of complexity. Some cells are growing really big, some are dividing. And so at the cellular scale, there's a lot of heterogeneity, but those sepals are growing uniformly. So we use the Morphographics image analysis program to track all of those cells in time, and we can track their growth rates, their growth orientations, all this kind of stuff. This is lineage tracking, so each cell had a color, and each of its daughters get the same color. So you can, again, see complexity. Some of these giant cells just grew throughout the whole time and never divided. And then other cells made these very complicated lineages. Okay. So I think of organ formation as a bit of an emergent process. You've got all of these cells interacting with each other, and you get properties at the organ scale that are not obvious from what the cells are doing. And this is like snowflakes, right? Water molecules are interacting at a very local level with no rules. The cells have rules. These are having just physical rules of interaction, no blueprint, and they make these complex structures. So I think that's a lot of what we're facing in biology is these emergent processes, and how do these local interactions in neighborhoods give rise to things at larger scales. Okay, so cells are highly variable. They're highly variable in size. They're highly variable in their division. Some cells are dividing a lot, some cells are dividing a little. This is a heat map with the daughter cell count. So the cells that didn't divide are in this deep blue purple color, and cells that divide a lot are in more like orange. And there's also highly variable growth rates. Some cells are growing faster than others, and they can be growing faster right next to their neighbors. So we've got a lot of variability here. And so that really brings up that question of, if we have so much variability, how do you get robust organs out of this? Okay, and this has agricultural relevance. So we're going to do an interaction here. How many, look at these two broccoli heads. How many of you would buy the red one? No one. How many is going to buy the blue one? Some people. Some people are just not buying broccoli. Yeah. I do like broccoli, but I know there's a lot of people who don't. Okay. So most people are going to buy this one and not this one, if they're going to buy broccoli at all, because this one, the size of those flower buds is not robust. And in case you didn't know it, when you're eating broccoli, you're eating all kinds of flower buds. That's what you're eating. So this broccoli is actually relatively closely related to Arabidopsis. We don't have an answer to this problem yet. It happens a lot on the East Coast that you get these irregular bud sizes, and it's because of temperature variation. So most of our broccoli is grown in California and shipped over, which is not good for our nutrition. These are perfectly nutritious. People just don't like the way they look. Okay, so yeah, so how do we figure out what makes robustness? And sometimes we're geneticists at heart, so we said, well, we'll take a mutant screen approach. And we'll say, okay, we've got our wild type flower. The sepals are the same size. If we want to know what controls robustness, we need to find mutants that lack that robustness. So we've got different size or shape flowers in the same plant. Yeah.
When you talk about the variability, do you also compare flower to flower? Like, do you say the same? Can you recognize the similar zones where you have large cells, or is it like really completely random? If you go from look at different flowers.
Yeah, okay, so across different flowers, the position of the giant cells is random. Okay, so we've done actually some cool new studies on that, because it's very hard to say something's random. And so what we found out is that when they're formed, the position is random, and as the cells divide around them, that gives them a clustering that becomes non-random if you consider them in the body of the whole cells. So it's actually a pretty cool pattern. But yeah, from sepal to sepal, it's random, and it's sort of diagnostic, because once they're there, they're always there. Mm-hmm. So when we're live imaging, we kind of use them as like, oh, we're on the right sepal because it's got this pattern of giant cells. Okay. So yeah.
The number of giant cells, is that a sort of relatively conserved thing, or is that also equally very variable?
So it seems like it's tuned. We've actually, I'm not talking about this today, but we did a lot of work and showed that the specification of giant cells seems to be, well, is from the fluctuation in concentration of a transcription factor called ATML1. And our models can reproduce the giant cell pattern based on just stochastic fluctuations. They also reproduce this random to clustered pattern, which is pretty cool. And we usually get like about 14 giant cells, but that's an average, and it can go up and down from that. So we think it's just tuned. Yeah.
Polyploid, the giant cells?
Giant cells are polyploid. Yeah. Mm-hmm. Endopolyploid.
And the polyploidy vary, or is it a constant number?
The polyploid varies between the cells. So we have everything 2C, 4C, 8C, 16C, maybe 32C. Yeah. And so the big ones, the giant cells, are the 16 and 32C, and like 8 is sort of a borderline. Yeah. Okay. So we're going after robustness today, not giant cells, because that's the way I chose. They're both equally cool stories. So yeah, back, now quite a while ago, my first postdoc, Leland Hong, did a screen for mutants with variable organ size and shape. And she pulled out these VOS mutants, which now have other names because once we figured out what the gene was, it had another name. And if you look at these, they're very interesting. So VOS1, or FDSH4 as we're going to call it, has different size sepals within the same flower. You can see it also has different size petals within the same flower, different size stamens. Then VOS2 has different size sepals. This is DREAMY1. And VOS3 is AS2-70, that's a typo. And it has variable shape. It has all these lumps and folds in the sepals, but they are the same size approximately. So yeah, go ahead.
And the mutant shape is robust again. As in, like, if you compare the same mutant across different plants, they always have a similar shape.
This one, they always, so it's not robust. Okay, we're going to come to that. It's not robust in terms of each sepal is a bit variable in what that shape is. It is robust in the sense that they're always messed up, okay? And these ones, in VOS1, they're not always messed up. Some flowers have normal sepals, and some flowers have all abnormal sepals, and you get everything in between.
Yes, but since they're not messed up in the same way.
In this one, they're all buckled,
Okay,
but the buckling pattern is different, at least slightly different.
got it.
Can we put it that way?
Yeah.
And we're gonna, what I want to do today, what I thought would be fun is to sort of reflect back on the lessons we've learned over the last decade and more from these mutants and sort of put together what have we learned about robustness and morphogenesis from these mutants. Because we spent a lot of time looking at these mutants. So yeah, we've kind of pulled out, I've framed it as these principles of robustness of morphogenesis from all of these studies. And so I'm going to go through these in order a bit. And so the first one I want to talk about is how spatial temporal averaging produces robust floral organ size and shape from heterogeneous cell growth. And this is really focusing on FTSH4, VOS1, and it's the work of Leland when she was in the lab, and more recently of my former grad student, Bella Berta, who's now at Princeton as a postdoc. And so if we look at FTSH4, we can see at early stages, it does not have the same size sepals, and the consequence of this is that the flower buds are open, whereas wild-type flower buds are closed. And in our growth chambers, that's okay. They're in great growth conditions where we keep the pathogens as low as possible, not zero, but low. But you know, now the developing reproductive organs are exposed. And so this would be a highly detrimental phenotype outside. I've already shown you that when they're in mature flowers, you end up with different size sepals within the same flower, whereas wild type's very robust within a flower and across flowers. Here, there's a lot of variability. And if we dissect off individual sepals and look at them, again, you can see these are the 4 sepals from a flower. And they're consistent size and shape. These 2 are the lateral ones. They're a little more pointed. But here, if we look at FTSH4, you can see they're definitely variable. Like this one is way smaller than the rest of them. So how does heterogeneous cell growth produce robust organ size and shape? When we were doing this, we were working with Arezki Burud and his grad student, Mathilde Dumont. And this was a Human Frontiers project, so a really fantastic collaboration. And so they started thinking about modeling this. And so they were modeling the idea that you have cells growing fast and cells growing slow within the same sepal. But when they model it, let me play that again. When they model it as each cell is growing fast or slow, it's choosing its growth rate from kind of a random distribution. So sometimes it's fast, sometimes it's slow. The neighbors are doing all kinds of different things. You can see that all of that fast and slow growth is essentially averaging out as the sepal grows. And if we outline those sepals at the end, you can see they're quite reproducible, despite the fact that they have a lot of heterogeneity in their growth rates in individual spots. Okay? And then the converse is that if you don't allow that variability over time, and you have fast and slow growing positions, those then really make what you'd expect. The fast growing really extends out, the slow growing really constrains it. And you get highly irregular shapes out. And so that one, you know, you can see all of these different irregular shapes. Yeah.
So this is just like the rate at which the volume is increasing, but it's not necessarily, how is it related to division time?
So in this model, we have no divisions whatsoever, and we actually don't have cells. It's a continuous sheet of tissue with triangles in it. And they're, the way we're controlling growth rate is actually through stiffness. So it's a finite element model. And so we're making the pieces stiffer or softer. Yeah. So we are completely ignoring the fact that cells divide. And we are actually, even within a cell, we did some work to see that the stiffness is not uniform within a cell. And so we're kind of disregarding cells altogether in this model. Yeah.
In this model, if you pick a very smooth stiffness across the plant, then even if it's not homogeneous, you wouldn't expect it to be to form a irregular shape, right?
Yeah, so if you had uniform stiffness or some like nice smooth gradient of it, then you do get regular shapes out. Yes. But so people keep asking me like, well, do you need this heterogeneity for anything? And that's something we're addressing now. We think it's giving the plant flexibility. And it may be sort of a byproduct of fast growth. It has to be sort of heterogeneous just because it's noisy, and then the plant has to accommodate it. But if the plant could do smooth growth, it would be okay. We just don't think it can.
What about the thickness? The other direction, how variable is that?
So sepals are about 5 cell layers thick. That's not variable. This is one sheet model. It doesn't account thickness at all, but we don't see variability in that until we get to the, when I show you the buckling one, we start getting buckles. Yeah. Okay, so they had this model that, if variability changes in time, or the stiffness changes in time, the growth rate changes in time, it should average out and be okay. And so we were live imaging at the same time going, okay, well, that's a theoretical model. We're going to see what happens in the plant. And at some point it came to us, oh, the plant is doing what they showed in the model. And it took us a while to get there, but that's what we found. So this is Bella's live imaging. If we start with wild type, so here we've got a heat map. Fast growth is in yellow, slow growth is in purple. And we've got the cells that divide outlined in white. So you can see where cell division is happening, where growth is happening. And we have this overarching growth pattern where the fast growth starts off at the tip and comes downward. And that's specified. That would be our smooth growth thing, is that you have this growth gradient. We call it the base of petal growth gradient. It's starting at the tip and coming downward. That's the classic morphogenesis of these things. But you can see that there's a lot of variability within that. That's not the main source of variability. We've got all of these different cells growing at different rates. If we look at FTSH4, we still have this same base of petal growth gradient, but it's a little bit altered. So you can see it does start off faster at the tip and come downward. What you can see is it's a little bit patchy, and we seem to have patches of faster and slower growth. So we've got this patch over here that's a bit faster, and this patch over here that's a bit slower, and that continues over time. Whereas in the wild type, you don't see continued growth, right? These cells are growing fast, and a little slower, then a bit faster, so they're not staying with the same growth rate.
Yeah, I was just wondering, how is the growth rate correlating with the cell sizes? Is there a correlation?
No, because, well, we don't see a correlation, or not an obvious one, and the cell sizes are getting bigger because of endoreduplication, so they're stopped dividing and they're growing, but when we've looked at that, they're growing at the same rate as the clone of dividing cells.
So on this right image here, bottom right, they're just, like, is that a segmentation artifact, or they're just like lots of small cells there, like that mosaic pattern?
Here.
Yeah.
No, this is real.
Okay.
There's lots of small cells right next to a giant cell, yeah. That is completely classic sepal, yeah.
Okay.
Yeah, so these cells, you can track this one backward. Okay, I'm losing it right around here. But we have, all of these cells are tracked over time. And so this one is just growing all that time, whereas you can see there's lots and lots of divisions going on.
Specific, you just see it.
No, that's a sepal pattern, not a mutant pattern, yeah.
How do you measure the growth rate? What I want to know, what is the time interval that you use to compare volume? Because if you measure, at a very small delta t, okay, it's more difficult.
Yeah.
It's more noisy.
Yeah, exactly.
To
Yeah.
extract the data is more difficult, and you have less temporal averaging. If you do it over a longer delta t, you smooth out the data just because of the way you do it. So I presume that depending on the delta t, you would have different results. Not that I mean that the noise that you see, is due to the way you measure, but nonetheless, there's something I would like to know a little bit more about. Maybe you could...
No, that's a really good point, right? And that's a sort of experimental thing where we have to find the right experimental value, because as we extend delta t, it is averaging. So what we've picked and this may not be the right choice, but like it's the best choice we can make, is imaging every 24 hours. And that does do some averaging. We've also imaged every 12 hours, and we get, we definitely get a little bit more noise when we have 12 hours. 12 hours also includes the noise of the circadian rhythm, which like adds another complicating factor we want to get rid of. But I mean, we sometimes do 12 hours. When we get down to 6 hours, then we start worrying about experimental noise outweighing growth noise. And so we've sort of picked this 12 to 24-hour window as being experimentally what makes the most sense. Yeah, and it's also, we can only have so much imaging of the plant before it starts affecting the plant growth, so we try to decrease that as well. The growth rate that we're measuring is a relative growth rate, and it's really a ratio. It's the later time point divided by the earlier time point.
Maybe the question is there any system, this one or another one, in which you could actually track, say cell surface or whole volume?
So we are actually doing
continuously so that you could get a sense of how much kinetics of growth is smooth or changing over time during cell cycle and things.
Yeah, we haven't done that. Charlie Anderson was going to look at that, and I don't know if he ever did that. But yeah, he was going to look at like shorter time over the cell cycle. So at longer timescales, we don't see an effect of division on growth rate, but at shorter timescales, there very well may be. And we haven't looked at that. I don't know if he ever did that, and maybe if he hasn't done it, we should jump on it, but we haven't.
And maybe my last question is, I don't know how it is in plants, but in animals, the density of the cytoplasm is very critically tuned. So, as you synthesize more proteins, the osmotic flux are adjusted in ways that are not fully understood. How is it in plants? Because of the vacuole, you could pump in a lot of fluid, increase volume in a way that does not dilute the cytosol.
Yeah.
So there's more free, leeway to increase volume in a way that does not reflect what I would call true growth of mass, dry mass increase.
So I completely agree with you. There's two types of growth going on in plants. There's vacuolar cell expansion, and I agree that is not true growth. And then there's, but it does increase size, right? So so we have to take that into account. And then there's cytoplasmic increase. We need to show this better, but we're actually looking at area, not volume. And part of the reason we're looking at area is because the volume is mostly taken up with the vacuole. And so surface area is a better representation, and we're just only looking at top area, of the cytoplasm. And so that's partly why we've chosen this like area as opposed to volume measurement, because of exactly that reason. That's one thing where the polyploidy, I think polyploidy goes with cytoplasmic volume. Because that's the one that's correlated, not vacuolar volume. And you'll see throughout the literature, people say, oh, the vacuole expands and polyploidy happens. No, it's going with the cytoplasm volume. But we're looking into polyploidy more, and we want to really pin down that relationship a little better. But so far, it looks like that. Yeah.
So these spread blocks are following cells that are moving along their trajectory?
The cells will stay in place. Cells don't move.
Oh, they don't.
So we're looking at how the growth rate transitions through the tissue, but all the cells stay in place. Plant cells are locked in by their cell wall, no movement, which makes our life easier because it makes it pretty easy to track them even when over fairly large time periods. Okay. So then we looked at averaging by looking at over 3 days. So if we look at growth over 3 days, then we're getting, you can see that there's still variability, but we're getting these sort of nicer growth gradients in wild type with more uniform growth. And if we look at FTSH4, so these, I should point out, these are 3 different replicates. So you're seeing 3 different live imaging over 3 days. And here, if you look at FTSH4, you can see it's kind of patchy. We've got this like slower growth and fast growth, slow growth patches. This one's more normal. Again, we have variability in FTSH4, so some are more normal. So that is suggesting to us that it really is averaging over time. And in wild type, and it's not averaging over time in FTSH4. Okay. So if we want to quantify the fluctuations, we need we've got two things contributing to it, right? We've got the base of petal growth gradient, and then we've got the deviation from the base of petal growth gradient. So this is in collaboration with Fritjof-Brons. It came out of our last KITP meeting. So we've got sort of what this smooth gradient is and how much the cells are deviating it from it in their growth rate. And so we can characterize those fluctuations. And so we've got that across time. And so he can measure the fluctuations. And if we look at so we're subtracting out the base of petal growth gradient and just looking at fluctuations from it. If we look at wild type, you can see the fluctuations are pretty scattered, and some cells are growing faster, some cells are growing slower. There's no consistent pattern to it. If we look at FTSH4, now we start getting these patches. We've got patches of slower growth, patches of faster growth in it. So wild type has it's uncorrelated heterogeneity, whereas in FTSH4 it's correlated. Come on.
So the other thing is spatial averaging. So if you were to coarse grain a little bit the field, say to encompass 4 cells on average in each of the little bins, do you then see more reproducibility? I mean, in other words, what is the length scale over which you have spatial averaging?
Yeah.
Is it a couple of cells? Is it more?
So Oresky has looked at this a little bit more theoretically and looking at length scales of fluctuations. And so he's looked at sort of the theory of like how long these fluctuations are. I'd have to refer you to that paper. But here, I don't think we've done exactly what you want, but it's a good idea.
The reason why I ask is, imagine that cells would locally, cells grew a little bit more than you ought to, then maybe there could be some kind of compensation by talking to its neighbors. So if that were the case, then you'd expect to have a length scale of just the length scale of communication amongst the cells.
Yeah.
If it was kind of a random, if you have a distribution of growth rates in a population that is random without any communication, it would be good to know what would you expect in terms of correlation, length scale of this averaging, I think as a reference.
Yeah, so we haven't done exactly what you want, but we have done, what we've done is sort of look at a patchiness calculation. And this is not, so when we did calculate just growth variability relative to nearest neighbors, we don't see what patchiness coming out because we still have these growth rate differences next to neighbors. So Fritjof created this patchiness that looks a little bit further distance. And that's where we do see this real difference between wild type is less patchy than FTSH4. But yeah, it would be, one of the things that Fritjof wants to do in the future is look at this negative correlation. So if we have negative correlation or random, it will average, but if it's positively correlated, that's where it inhibits averaging. And he has found some evidence of this negative correlation, and we would expect to see that, but we need to dig into that more. And I think he's going to go after it in this data set. So then we looked at time. I think our data keeps going back and forth on whether time or space is more important. So some data sets, it looks like spaces, some looks like time. Let me just get through this first. So if you look at wild type and we look at the growth rate at fluctuations at early time versus fluctuation at a later time, in wild type you see they're not correlated. But in the mutant, they are correlated. So it's correlated over time as well. And I think that could be part of the big problem here. So it's temporally uncorrelated growth fluctuations in wild type, temporally correlated growth fluctuations in FTSH4. And again, this is looking over time at these different sepals. And you can see, it's got all these growth fluctuations, but wild type is very regular. And in FTSH4, you've got this side growing faster than this side. And you can see that side is, kind of making a tip, and this slow side is lagging behind. So we do see a correlation with it. Yeah, you had a question.
Extremes of the growth rates in the two cases, are those related or not? Is it only the shape of the regions that is different, or are the extremes?
The extremes seem to be the same. Yeah. At least at the early time points, later on, FTSH4 growth rate goes lower than wild type. But at these time points, we're looking at, so around here, well, they're not too different. After this, FTSH4 grows slower than wild type. But at these stages, the fluctuations are the same.
Understood, but do you look at anisotropic growth fluctuations? Because it also looks like there's some difference in orientation and elongation of the cells.
Yes, we have looked at that as well, and we see exactly the same thing. Wild type averages better in its anisotropy than FDSH4 as well.
Can I ask a related question? So when these cells are dividing, they are surveying the force balance around, right? And which axis to orient the cell division in. So when it's heterogeneous, is it not happening? Is it so slow that they don't care about their neighbors? If this temporally correlated with fluctuations, there are regions that start to do more and more oriented division then. Does that make sense? Like they correlate because all the cells are trying to divide in the same axis.
So it doesn't, we don't see major, I mean, division is affected somewhat, but so division in plant cells, I mean, it does respond to stress, right? The orientation is responsive to stress, but it's also responsive to the geometry of the cell. So these two things balance out. And we aren't seeing major defects in the orientation of the cell divisions in FDSH4. They seem reasonably normal. In cell division is just laying down a new wall. They don't like round up or any of these things. And so it is responding to force, but we don't see a huge defect. Maybe there's something subtle there that we should look for. But because we should see more stresses like between these slow growing parts and these fast growing parts, it should be generating stress.
Defect, I'm talking about just more bias.
Towards something, yeah. We haven't found it yet, but maybe it's in there, so that would be cool. We could look for it. Okay, so to summarize this part, and we're going way more slowly than I thought, so we'll see, which is great. We're having good discussion. So wild type has uncorrelated fluctuations and they average. The mutant has correlated fluctuations and they fail to average. So what is FDSH4? It's a mitochondrial localized ATPase protease. It's on the inner membrane of the mitochondria. What's it doing? It's generating, well, normally it's doing garbage collection. It's making sure the integrity of the mitochondria is good. And so when it's defective, what we see is that FDSH4 accumulates too much reactive oxygen species. So here's a stain for superoxide. You can see wild type, it only accumulates at late stages. FDSH4, it's there at earlier stages when the sepal is developing. We look at hydrogen peroxide, same thing, much more and in much earlier stages. And so then the question is, well, is this causative of the phenotype? We can reduce reactive oxygen species by overexpressing catalase. That restores robust sepal development. So it really is the reactive oxygen species that are the problem. Toman, did you have a question?
Maybe related to this, if you say in the mutant, what we see is there's a correlation between correlation in the fluctuations and the aberrant size of the tissue. And so that suggests that in the mutant, unlike in the wild type, there's some kind of aberrant signaling going on that coordinates things that ought not to be.
Yeah.
And so if that is the case, a suppressor screen would identify whatever cascade of signaling pathways to that. But I think it really.
Yes, we've got one suppressor, but I think it would be very interesting to do a suppressor screen. Things that do reduce ROS, we've got a number of ways of reducing ROS, and they do rescue. But there may be other things in that cascade that are super interesting. Yeah, I think that would be fun.
Do you have a sense for what sets the spatchiness length scale?
No, we don't. I think that's a super interesting question. And so far, we would love to know if it's like set by patches of ROS. We have yet to find a ROS fluorescent marker that works in this tissue at the right level. Like we've tried everything and we can't find it. So this is a low level of ROS, and most of the ROS markers sensors go for high levels of ROS, and we're not in the right dynamic range. And so that's why we're doing the staining, because we can stain for longer and get down to that lower level. But if we could find the right ROS sensor, I would love to look at that.
Sorry, just clarifying. So where is the ROS within the cell? Does it also diffuse extracellularly or all contained within cells? Or?
So we think it's diffusing extracellularly. So hydrogen peroxide particularly can go through aquaporin channels, so it's probably going out. You can see with the hydrogen peroxide, it seems to be more uniform across the sepals. With the superoxide, then we see these sort of patchy patterns that may relate. The problem is we can't get growth out of this, but it might relate to the patches of growth. And we also see, interestingly, in wild type, it does come on from the tip to the base, coming back to it may be a real signal in wild type that then is essentially overexpressed in the mutant. Yeah.
Since everything in plant converges at the end on auxin, and since auxin is kind of involved in growth, at least its orientation, my question is it also involved in the magnitude of growth?
Can I answer? It depends. So there is a paper here saying that this all relates to auxin, and they put auxin on and it rescues. We cannot repeat that. We think their allele is, we think they have a secondary mutation in their allele. So far, we haven't found the relation between this one and auxin. It's got to be there because auxin does everything, but we haven't found it yet. The next one, we're going straight into auxin. If we, yeah, we can get through a little bit of the next one. Okay, so we found that spatial temporal averaging produces robust organ size and shape from heterogeneous growth. We've also sort of found that hubs and networks like ROS, right, it's a real signaling hub, are essential for robustness. And so when you start disrupting hubs and networks, you start getting defects. Okay, so I'm going to go into the next point, which is timing is crucial for ROS morphogenesis, and it's going to come to this hubs and networks, and we may not get to the buckling at all at the rate we're going, but that's fine. Yeah.
I'm a bit surprised. There was, I don't know if you know, there's this work by Maha and Salem about if you have a growing elastic sheet and you have just random noise, you'll always see instabilities, where if it is correlated on larger scales, you can guarantee flat growth. And it seems like you see the opposite effect here.
Yeah, isn't that interesting?